Thank you for allowing me to be here to speak with you this afternoon. As Ethan mentioned, I'm Dominique Pichard, and I'm the Director of the Division of Rare Diseases Research Innovation at the National Center for Advancing Translational Sciences, or NCATS, at the NIH. We've already spoken about this several times today, and so I really don't have to dwell on this. You know, the challenges with rare diseases. There are more than 10,000 known rare diseases based on science and technology today. That number will continue to increase. And as you've heard already today, only about 5% of those rare diseases have treatments. There's a tremendous gap in getting us to every individual affected by rare diseases with treatments to change the course of their disease, to give them an improved opportunity for life without their disease. The other challenge that we haven't really talked about today is the time that it actually takes. So once you make that discovery in your basic science lab, the black box from getting there to your FDA-approved treatments. And that is really where NCATS spends a lot of its time, in that translational science. We need to make these discoveries actually into treatments, and that is a tremendous period of time. You see, on average, it takes, with our historical data of kind of the traditional drug development, 10 to 15 years from that discovery until it's in your medicine cabinet or the infusion in the hospital that you're able to get. And 9 out of 10 of those clinical trials, promising therapeutic candidates, will fail. Huge failure rate, tremendously long development timeline. 95% of rare diseases don't have treatments. So the NCATS mission is really to turn these research observations into health solutions through translational sciences. How can we improve the process? And as was mentioned, the vision at NCATS is to have more treatments for all people more quickly. And really we work to re-engineer that translational pipeline so that there are roadblocks that we know about within the translational pipeline, that 10 to 15-year time development. How can we smooth out those roadblocks, create efficiencies, find commonalities so that we can get to our end goal of more treatments more quickly? And what is our approach? So really, these are kind of some highlights of our approaches, but really importantly is that first area, understanding what's similar across diseases. With more than 10,000 rare diseases, if we go one disease at a time and we reinvent the wheel each time, we all do the same thing. For every single one of the diseases, we have to understand the disease. We have to get a detailed natural history. We have to develop clinical trial endpoints, so these outcome measures or biomarkers that was just being talked about for every single one of these diseases. That's not even talking about developing those therapeutics. So that's not talking about each individual drug, what has to go into, or gene therapies. So we really want to understand what is similar not only across diseases, but across the mechanisms, the processes, so that we can create efficiencies. We want to develop models that better predict a person's reactions to treatment so that we can lower that failure rate. We want to enhance clinical trials so that the results more accurately reflect the patient population. And growing more and more, we see the need to leverage real-world data and data science approaches to address the public health need. Rare diseases really have a home at NCATS. I think rare diseases exemplify a lot of the translational science challenges and issues. And by approaching those roadblocks in rare disease research, there's a much broader impact in translational science. And so NCATS really develops new ways to understand and treat rare diseases. And we do this in, I'll say 3 broad buckets. We create innovative opportunities that provide funding to advance medical research. We try to use data-driven solutions. And again, this will be increasing as more data is available. When you have rare diseases and there's not much data available, it's challenging to have data-driven solutions. So we need to create these data-driven solutions, create the opportunities to have them. Really importantly is the resources for our patient and patient advocacy community. You've already heard this afternoon, patients are part of the solution. Patients, their caregivers, their advocates, they are sometimes the ones creating the solutions. We need to make sure that we are involving them in the research process early. So at NCATS, we spend quite a bit of our efforts working with the patient community, creating opportunities to educate them, to empower them, so that then when they are moving forward with building foundations, raising funds, that they know what to do and how to do it, so that we can all move forward together. I'm going to tell you about a few of our initiatives that really exemplify some of the work that I've been talking about and what our approach is. So I'm going to start with the Rare Diseases Clinical Research Network. This was established in 2002, in the Rare Disease Act of 2002. And this program was initiated in 2003, and it's recompeted every 5 years. So we are currently actually have a funding opportunity out to initiate the next cycle. What this did is it allowed us to create this research network of individual consortium for rare diseases. And this next slide really highlights our current network. Each one of these is a different consortium that has to include at least 3 rare diseases. So again, we're going more than one disease at a time. We need to find the commonalities. As you can see on the left-hand side, the key stakeholders are the NIH, and along the bottom are the different institutes that work along with the National Center for Advancing Translational Sciences. So we fund these opportunities. There are the researchers and the clinicians who have applied and are running these research teams. Every single consortium has to engage the patients and the patient advocates group. They are part of that research team. And that is really important because, as you've heard, the patients have to drive the solutions. They know what is most important. They know how to improve that feel function. Survival is a little bit more clear. But the feel and function, that has to be driven from the patients. They can guide what's important in this research. And so each individual consortium, as I mentioned, has 3+ diseases. But in this network, I've seen it. I've been at NCATS for about 9 months so far, and I've seen this group engage. They are learning from each other. What one group develops as an outcome measure and presents it to the group, the next group says, we have similar issues in our disorders, our group of disorders. So now we're amplifying. The work that is funded for one group really can have a much broader impact. And really what we want is to get treatments. And this slide highlights the translational impact in terms of actually getting treatments to patients. So we have studied across the years, across the cycles. There have been about 200 rare diseases that have been studied in the RDCRN. We have developed the infrastructure. So they all have a natural history study. They usually work on some other clinical trial, outcome assessment, development, or biomarker assessment, which really then lays the groundwork to be able to get commercial interest for developing therapeutics. And what this slide is showing you, the 12 FDA-approved treatments for 11 of these diseases. And it's not to say that these were pivotal studies funded by the RDCRN, but rather RDCRN did all the hard work, allowed these diseases to be ones that could be of commercial interest, and then pharmaceutical companies brought this across the finish line. Now, while 12 does not seem like a very large number when we're talking about more than 10,000 rare diseases, I think I'm very encouraged by this when you see the dates of the approvals. 5 of them have been in the last year. And that also goes to show why this process, it really demonstrates how this process can be quite slow. You have to do all that groundwork. It can take quite a bit of time to understand the diseases well enough to develop those outcome measures so that you can have successful clinical trials. And by setting up this infrastructure, the hope is that you then don't have the pharmaceutical companies that end up closing programs because you're more likely to have success. I'm going to transition over to some of the programs that we lead. This is really just to give an overview of what I'm going to be talking about for the next 2 programs is gene therapy. In this very oversimplified view, you have a gene that has an error in it. And I will say that about 70 to 80% of rare diseases have a genetic mutation, a genetic origin. And so this is really for the majority of rare diseases. So you have a gene that's faulty. You want to replace that faulty gene with a correct gene. And so through this AAV gene therapy, you use this viral vector to deliver the correct gene to the cells of the body where they need to go. And then, we'll call this delivery box, gives you the corrected gene, and your body does what it needs to do to make this into the corrected protein. So really oversimplified. But what it's doing is getting to the root of the issue. And so this really is something that for the genetic diseases is of high priority. So two programs that really look at this gene therapy are the Platform Vector Gene Therapy Program. And so this was really meant to say, how can we create a system that will ultimately be a plug and play for all disorders that come down the road? And the idea was, let's lay out the framework. Let's solve many of the questions that are associated with gene therapy. It is quite complex to do the manufacturing because your little delivery box is actually different for each disease. It's not just the gene itself, because you have to get it to different places of the body, and you have to have it maybe regulated. Too much or too little can be bad. And so the concept of the platform vector gene therapy is to do this so that then when other companies that have gene therapy products come along, they use the learnings that were made. And so this program really will share all of the learnings from the interactions with the FDA. So what is it that was done? How did we learn about how this process needed to be from the FDA? What feedback did we get? And all of that will be publicly available to de-risk it for the next groups. So this is, one of the milestones was the PAVE-GT successfully navigated the Food and Drug Administration orphan drug and rare pediatric disease designations for this particular gene therapy. So there are several candidate gene therapy programs through this. And so each one, we're learning each time we go, and we'll be sharing the learnings so that the pharmaceutical companies or patient advocacy groups that are leading these efforts can learn from our successes and our mistakes. Next, just to touch on another gene therapy program called the Bespoke Gene Therapy Consortium. This is a public-private partnership, and the goals of this are, again, to make this AAV technology more accessible by optimizing this production and sharing the learnings of this. So not only are we optimizing it for the particular diseases that are being tested in this consortium, but sharing the learnings. We want to streamline all of that work, that 10 to 15-year process, so that the preclinical and product testing is really much more streamlined. I will say gene therapy is relatively new kid on the block compared to small molecules, and so there's a lot still to be learned about the process for the early phase testing, that preclinical, as well as the product testing. This next slide shows the public-private partners that have been engaged and the commitments. And so this has really been a huge lift and tremendous partnerships that have allowed this program over the last almost 2 years now to continue to grow, and hopefully we'll soon be talking about the clinical trials for these diseases. I'll actually skip through. So this is the portfolio of the diseases that were selected. This was an application process where researchers and patient advocates together could apply to be selected as a disease. All of these diseases had to be ones of no commercial interest. That was the goal. We want to do this to show how we can take diseases, really small populations, really don't have that infrastructure yet, are of no commercial interest, and still have successful gene therapy trials so that the next ones that come along can follow the playbook. And in fact, we have released a playbook. This was the version 1.0, which was released last month at the Accelerating Medicines Partnership annual meeting. And this is a playbook that helps the research groups and the companies in gene therapy just understand the process. It just simplifies it and tells them kind of how to put one foot in front of the other for this gene therapy, And as we continue along this pathway, we will iterate and create new versions of this playbook with new learnings. And finally, I want to talk about one last program, somatic cell genome editing. And so gene therapy is really exciting, and it is the area of genetic medicine that has been around longer. And so there's quite a few programs that are coming along, but the next set of technology that's exciting is gene editing. Again, this would be useful for the 70 to 80% of rare diseases that are of genetic origin. And so this somatic cell genome editing program is intended to develop novel technologies to build that foundation for clinical application, and then deliver the resources to accelerate treatments. So in phase 2, we are actually looking to find better ways to actually get those editors to really challenging organs such as the brain. And so there have been these challenges, these prize challenges, where we seek to get the most innovative solutions for this significant challenge of genome editing. How do you develop this technology, and how do you deliver it successfully to the organs that need to be delivered? And I think in all of these, and I'm going to skip ahead to this one, the underlying goal in NCATS is to, as I said, our vision is to get to more treatments more quickly for all people. And the only way we're going to do that is if we work many diseases at a time. So we need to find those commonalities. We need to see where do diseases have overlap, either in their molecular etiologies, in the approach to their treatment, or in the symptoms. How can we look across the diseases, find those commonalities? And how do we develop therapeutic platforms, such as this bespoke gene therapy or the somatic cell genome editing, that can ultimately get us to those treatments faster? Thank you.